Thank you very much, and good afternoon. I'm acutely aware that I'm the barrier between us and lunch. I hope that a few of you had a sandwich or at least a cake or something so that you can keep with me for the next 20 minutes and then questions. What I'm going to talk about is a very, very simple but highly effective technology that Angle has developed and owns. And this technology has the potential to actually revolutionize the way that we treat cancer, because what it allows is the accessing and harvesting of cancer cells from patient blood for analysis. This is crucially important because it means that you can then tailor the individual patient's cancer treatment to their particular cancer. So that's what I'm going to be talking about. It's all about precision medicine. And there we go. And the background, obviously, with cancer is, unfortunately, not only do we have a lot of cancer presently, but this is growing. And Cancer Research UK have told us that people born in the 1960s and onwards have a 1 in 2 chance now of getting cancer. And that requires us to be very smart in how we treat that, to try and reduce the symptoms for patients, give them better outcomes, but also to make sure that we spend money in a responsible way. And our technology can help with all of those different things. One of the major problems with cancer is that it is personalized to the individual patient. So you have several people with the same cancer in the sense of if they had lung cancer, you would imagine they had the same disease. But in point of fact, they don't. And that is where the drug companies are trying to recognize that and look for particular mutations which might exist within the individual patient's cancer, and then make a drug which actually targets that particular mutation and gives an outcome for those patients. So if you look at this example here in breast cancer, the Genentech drug, Herceptin, which is obviously a very well-known drug, only actually works for women who are HER2 positive in their cancer. The women who are HER2 negative, they won't benefit from that drug. And so obviously it makes sense to ensure that the patients who can benefit from the drug get it, but the patients who do not or cannot benefit from it because they're HER2 negative do not get that particular drug. And that's what we're talking about when we just talk about personalized medicine or precision medicine. How do you actually do that? Well, at the moment, it's all to do with the biopsy. Solid biopsy is where you cut out some material from the patient's cancer and take the tissues which are cut out to the biological lab, and they're investigated, and they look at the cancer cells. And in the case I just described, they look to see if that patient is HER2 positive or not. However, there's some issues with the solid biopsy. Some cancers, it's quite difficult to access the tumour, and therefore you can't get to the material. And in any case, once you've cut out a lump, it doesn't exist anymore. So you can't go back and cut it again because it's no longer there. And this is crucially important because the cancer changes over time. Becomes different. So whereas a patient might be HER2 negative on presentation, it's been shown that about 25% of those women will become HER2 positive later. But unfortunately, it's very difficult for the doctors to identify that and give them the drugs that would benefit them. So what we're pioneering is a liquid biopsy approach, and this is benefiting from a biological phenomenon, which is that a primary cancer tumour, the original cancer that patient has, will drop cancer cells into that patient's bloodstream, and they'll circulate, and they're known as circulating tumour cells. Those cells are actually very, very important cells because they're the ones that can land somewhere else and cause a secondary cancer. So that's how a patient can start with a cancer in one part of their body and then find that they've got cancers in other parts, known as secondary cancers, which of course are the cancers which usually kill the patient. It's the secondary more than the primary. So those cells, these cancer cells, circulating tumour cells, are existing in the blood. And of course, they would be a fantastically useful source of cancer cells for evaluation to make these determinations as to which drug would be effective and which would not, if you could get hold of them. And we've known for probably 15 years that these cells exist. And numerous companies have had a go at trying to get hold of them. But it is only now with Angle's Parsortix System, which I'm going to tell you about, that we can actually routinely and effectively get hold of those cells in a form that can be analysed. And that's what I'm talking to you about today. And we've got a consumable product. So basically, it's a razor blade model. I'm going to show you the instrument that runs this, but This is the actual one-time use consumable. It's a little piece of plastic. It's shaped in the size of a microscope slide, and that's obviously so you can look at the cells that are captured inside there if you want in a microscope. It's made from polycarbonate. It's going to be mass-produced. Obviously, you need a high skilled manufacturer. In this case, we have a nano manufacturer called Thinks in Germany who makes this. But we can scale this up. We can make tens, if not hundreds of thousands of these consumables if we want to do so. And what I'm going to do is show you how it works with this animation. So if this works now, we're going to see, whoops, it didn't work. Jump to the next one. Right, here we go. So that's the inlet port, and that's the outlet port, and the blood's going to flow inside of this cassette. So what I'm showing you is an animation. So those loops have got a series of steps, and the blood will go up those steps and then through a tight gap here. And the red blood cells are these smaller ones, the larger white blood cells, and these are illustrating the cancer cells. So you see the cancer cell cannot get through the step gap, the critical gap size. And it's as simple as that. So often when I go to medical presentations, I find it difficult to follow exactly what's being discussed. But our technology genuinely is as simple as that. And you kindly said that I was an engineer, which I am. I have an engineering degree from Cambridge. And that's the fundamental part of our technology, is an engineering understanding of how these things work. I'm now going to show you actual real blood flowing inside our cassette. So this is real blood flowing, and that's densely packed red and white blood cells streaming across the critical gap. We're looking at it from above. Those lines are the steps, so they're going up the steps and then through this gap. And obviously it's lighter coloured here because there's only one cell deep. And this is a trench of blood going out of the cassette. So it's just flowing straight through that little cassette I showed you. And I'm going to show you what happens when a cancer cell comes. Remember, there might only be 1 in 1,000 million. And that's one there. So that cancer cell has been captured on our step. They're always caught here. This is the critical gap step. And it just stays there. And then when the separation is finished, you end up with these large cancer cells trapped inside our device. And the great thing about that is that we can actually get the cells back out of the system. And the way that we do that is, here's our instrument, and that's a standard blood draw tube. That's an EDTA tube. It takes about 10 milliliters of blood, so a fingerful of blood from a peripheral vein. The user simply takes the top off that tube and attaches it to our instrument and then hits the start. And the machine does all the rest of the work. The user can disappear, and about an hour later, the 10 milliliters of blood will have progressed through the cassette, which is inside this clamp, and be in the waste, and the cancer cells will be stuck in there. So there's the consumable. It's sitting inside the clamp. Notice how we've got beautiful engineering that brings the microfluidic tubing exactly in the right place. And then the user can do two things. I said it's in the shape of a microscope slide, so if they want, they can put chemical reagents in here and stain the cells for positive identification, or to look for markers like HER2, and then take the cassette out of the clamp and put it straight on a microscope, and that process can be automated in terms of image capture and counting of cells. Or, and this is where our technology differs from most other technologies which are even a dream in somebody else's mind, we can change this valve positioning into the reverse, and then we hit the machine button again and reverse the blood out of, reverse the cells out of the cassette and collect them in a little tube. So then what we've got is 200 microliters of liquid, and inside there are the cancer cells. So in that 10 milliliters of blood, there might be 50 cells, 100 cells, something like that. The whole point of this is it's a very versatile tool that can then be taken and used in the same way that the solid biopsy material can be analysed. So you'll see more examples of that as I go through, but all of the existing molecular biological techniques that we can use on solid tumour biopsy, you can use on these cells. Next generation sequencing, PCR, FISH, and immunohistochemical staining, amongst others. Hidden down here is a very important point. We own this technology. That's absolutely crucial. It can't be copied. We own the use of the series of steps to separate particles from liquid. 2 granted US patents, and we've got patents being pursued worldwide. We should hopefully have a European patent fairly shortly. So what are we actually doing with this? Well, now that we've developed it, and we've actually spent quite a bit of time perfecting it, when you get these kinds of systems, it all seems like everything's perfect, but it's only when you've had the system out with end users, major cancer centres doing work for quite some time that you work out what are the little bugs that need to be improved. And I'm delighted that we've spent about 18 months doing that. We've now made this very reproducible. It's very consistent, it's a very robust system. And so what we're now doing is selling it. And the commitment we made to the market is that we are looking for our first research use sales for this product before the end of this calendar year. So as you can imagine, we're well down the track in doing that. And we've got several customers who are currently evaluating product, and so that's very encouraging. The scale of the research use market, we estimate, is around £250 million. The biggest component part is the different cancer drug trials. And we estimate that if we took 5% of the drug trials market, we could secure an £8 million revenue just from that. So obviously, this is not a market to be sneezed at. It's important, and we want to see some cash flows coming through from this. However, the research use activity is fundamentally important to our overall remit, which is twofold. One is to get clinical use of this product. So we need the big cancer centres to work with it and to find out the best way that they can benefit patients from the information they can get from these cancer cells. And the second thing is, in terms of the pharmaceutical drug trials, is that those people, basically we want them to be paying customers, but the reason they want to use our system is so that if they get their drug through approval, they can use it as a companion diagnostic. So the idea is our system will be used to identify which patients may benefit from which drugs, and that's a companion diagnostic, and then also to assess how well they do actually benefit once they receive the drugs. As I say, the research use market here is very, very significant for us for our future growth potential. And the research use is happening as we speak. Okay, so clinical applications. We estimate that the cancer patient treatment market is worth £8 billion per year. We've chosen one specific application. This platform works on a wide range of different cancer types for a wide range of different functions. But what we want to do is to have one specific application that we develop initially so that we can focus our efforts. And we're doing that in ovarian cancer, and it's specifically in relation to triaging women who have an abnormal pelvic mass. So what do I mean by that? Well, there are about 1 million women in the United States and Europe every year who have a growth in their pelvic area. And that this can be diagnosed through CT scan, ultrasound. So the doctors can identify that there's, in some cases, quite large growths from the ovaries or in that area. And those women, some of them will be elected to have surgery each year. So the doctors then start the operation to cut out this growth. But the problem is they don't know, are they dealing with a patient who has a benign growth, which, for example, might be a hysterectomy, and that would require maybe an hour's time from a general surgeon. Not particularly risky. I mean, obviously, it's not something you'd want, but you're likely to have a good recovery from that kind of operation. Or about 15% of the women will have ovarian cancer. And those women have got a very, very difficult situation because if they don't have a specialist cancer surgeon working for 6 hours and then go straight into intensive care, they will die. They will have a dismal outcome. A much worse outcome than if they hadn't even had the surgery in the first case. So the question is, who are these women? And at the moment, the test to investigate that, which are these ones here, CA125 is a blood-borne marker, which is like PSA for prostate. But unfortunately, its sensitivity is quite low, so it misses half of the women who have ovarian cancer. Or there's another product called OVA1 that has better sensitivity, but unfortunately its specificity is low. And what do I mean by specificity? Specificity is patients who are healthy and don't have cancer, can we correctly identify them? So if you only have a 50% specificity, then you are repeatedly saying that women who are benign are actually malignant and getting the wrong answer. And obviously that's as bad as saying that people don't have cancer who do have cancer. So what you need is something with a high sensitivity and specificity. And that's what our partner, the Medical University of Vienna, has done with our system. They've taken a simple blood test from women who have got ovarian cancer, and then they have used our system to extract the ovarian cancer CTCs, the circulating tumour cells, and then they've tested those using standard PCR analysis, which is an analytical platform that sits in all of the major hospital labs around the world. It's a pretty standard technology. And they've found RNA markers to identify those ovarian cancer cells with a very high sensitivity. So what they found is, first of all, they said that we've got 100% specificity. So you don't get ovarian cancer cells in your bloodstream if you've not got ovarian cancer. So we've got a very, very high specificity. We don't have false positives. And then with 7 RNA markers, they got around 80% sensitivity. They then increased it and looked for more RNA markers and got closer to 100%. So we think that we have the potential to use our Parsortix system for a simple blood test for women who are known to be having an operation, just to discriminate. Are you at high risk from ovarian cancer or low risk? And this is a market that's worth over £300 million per annum. And we believe that we have a tremendous competitive position in this because existing technologies don't work. And indeed, those people who are developing a circulating tumour cell alternative approach to us, they can't catch ovarian cancer cells at all. So that is an area where we've got a clinical study in progress, and the target is to complete the European end of that by the end of 2016. And that would then enable us to make sales in Europe straight away. And then following that, we anticipate the US side being about 6 months later, so middle of 2017. So we've had a lot of news recently, and some of it relates to multiple other cancer types. So Bart's Cancer Institute, just down the road here in London, they've been working with prostate cancer with our system. I should have said, by the way, on the ovarian, 100% of patients we got circulating tumour cells for out of their blood. And this was followed up here with 52 patients from Bart's in prostate cancer. We got 100% of them. We managed to get circulating tumour cells. This is unheard of, and we're changing the paradigm about circulating tumour cells. It's always been said it's technically too challenging, it's too difficult, you don't get enough cells from enough patients. But now we're getting cells from all the patients, and that changes the whole paradigm. And what we found is that Bart's discovered that not only did we get epithelial-based circulating tumour cells, but we also got mesenchymal-based circulating tumour cells. And they found that those mesenchymal cells were the ones that actually caused a secondary cancer. And the number of those cells in our system correlated perfectly to the patient's clinical condition. So the more cells, more mesenchymal circulating tumour cells, the worse the condition of the patient and the further advanced. So they're now looking very hard as to how they can find molecular biomarkers on those mesenchymal cells so that they can deduce which drugs will be most effective for prostate cancer patients. Moving on from that, we had an announcement, it's probably only a couple of months ago, from another one of our partners, University of Southern California, in the area of breast cancer, and particularly metastatic breast cancer. Now, they have so far a smaller number of patients, but again, 100% of the patients they got these circulating tumour cells from. The reason there's a smaller number of patients here is actually quite interesting. It's because the task that they were trying to do with these cells is particularly difficult, and that was to correlate the whole genome of the cells that we found in our blood sample through Parsortix with the whole genome of a biopsy from the metastatic site for the breast cancer patient. So they had breast cancer patients who were in later stage disease. They got secondary cancers, for example, liver cancer, and they then got cells from the metastatic site by solid biopsy. And that's why there's a small number there, not because of our system. It's simple to take blood tests from women with breast cancer, but it's not easy to do a liver cancer biopsy. That's a full surgical intervention to cut out part of the liver. But what they found was, and this is a sort of complicated scientific representation, but basically what they found was that on the circulating tumour cells from their blood test, they got all the genomic information that they got from the metastatic biopsy. So suddenly, instead of these surgical interventions to cut out pieces of liver and other very nasty investigative work that's needed to get these cells to work out are the patient's HER2 positive or negative, they're looking at the prospect of a blood test using our system. A simple blood test, which is what everybody wants. Now, the interesting thing was that our system found not only the genomic information from the metastatic site, but some other information as well. And so we asked them, the researchers, about this, and they said, but that's what we expected, because this patient has not just got one metastatic site, she's got multiple metastatic sites. She might have cancer in her bones, she might have cancer in her lung, she might have cancer in her brain. And the thing is, we can't get to those places to cut out any cells. But your system is capturing cancer cells in the bloodstream from all those locations. And therefore, it gives a lot more clinical information than you might get from a single metastatic biopsy. We had an announcement this week, actually, last week, which heralded the work of basically nearly 3 years by Cancer Research UK. And it was from the Manchester Institute of Cancer Research UK. Now, they are the research arm of the Christie Hospital. The Christie Hospital in Manchester is the biggest cancer hospital in Europe by number of patients. And Cancer Research UK Manchester Institute have been working with our system for 3 years. They have 5 systems installed. They have a full-time technician working with the system. And what they found, and they reported on, and I'd urge anybody who's interested to have a look at Angle's website, because the full publication, which was published in the Royal Society of Chemistry's journal, Analyst, is on our website. Incidentally, this presentation is also on our website. I'm sorry I didn't mention that at the beginning. It's on our website under the corporate presentations. But they found that our system, again, it worked with 100% of patients in lung cancer. It's suitable for 4 days at room temperature. You can take the blood and you can use it in a normal routine lab situation. If you look at some of the cell-free DNA things that people talk about, often that has to be processed within 15 minutes to 2 hours of blood draw from patient. That's practically very challenging, whereas Cancer Research UK said we're okay for 4 days. It's simple plug and play. I can't emphasize that enough. You put the blood in, you push the button, you go back and do your other work and come back again in an hour. It's not one of these things where you have to have half a dozen PhDs fiddling around. And it also enables the extraction of plasma for cell-free DNA. So lots of research organizations have started doing cell-free DNA analysis, looking for fragments of DNA in the blood. The great thing about this is that it's compatible with our system. You can use one blood sample for both. So every research group in the world that's doing cell-free DNA, they should be using our system as well with the same blood sample, and then they can get more genomic information than they can from cell-free DNA. And it enables analysis of CTCs not detected by epitope-dependent technologies. That's just a technical term, but basically it captures all of the necessary cancer cells. It's a bit like the one I mentioned with prostate earlier. And the quote that was Jeb Brady made, the Parsortix System has a unique combination of features, making it suitable for routine clinical analysis. That's the emphasis that I would like you to take away. We're trying to change the paradigm for cancer care worldwide. And this is supporting evidence in relation to that. I'm more or less up to my time, aren't I? So basically, our technology can be used widely with multiple different cancer types, as I've described. We have no change to the cassette that I showed you, no change to the instrument. It just works for lots of different types of cancer. And it can be used for lots of different things. We've already seen that it can be used for detection of cancer, and the example there was ovarian cancer. We've seen that it can be used for therapy selection, and work's been done in prostate cancer in that regard, and breast cancer. And it can also be used, we haven't really talked about it, but for remission monitoring. So patients who have had cancer, they've been treated, and now they need to be monitored. You could just do a simple blood test to see that they're not getting a relapse. So there's lots of different things that can be done with our system. And as a result, we've got a lot of different things coming out. So we expect a lot of news flow and results from the key opinion leaders, patient studies. In terms of sales, as I mentioned, we're working on that now. Cancer drug trials. We've had 2 publications in the last 2 months, but I'm expecting some more coming through and so on and so forth. FDA, we're in process with FDA, and we're expecting to have that come through fairly soon, I hope. Medtech deals, pharma deals. So basically, that's the last point. We have a system that can capture cells for analysis. We're not reinventing the wheel about how you analyze those cells. But of course, there are many large companies who have some fantastic analytical platforms, and they'd love to get their hands on the cells. And so we're opening up to collaborations with them. And so that's the summary slide. We've got 9 different major cancer centers that are key opinion leaders around the world. We've got a very clear path to commercialization. We're working on our first clinical application, and we've got high performance in multiple different cancer types. Thank you very much.
Thank you, Andrew. Thank you. I'm sure we've got some questions out there, but I'll kick it off. Because you said that what you really need is for the big cancer centres to start working with this technology, which you said yourself is a game-changing technology.
Yes.
So what's holding them back?
I can tell you more about that. So we've got some of the biggest cancer centres. One I haven't mentioned is MD Anderson in Texas, and they're working with our system in colorectal cancer. All of the cancer centres around the world know that the way to improve patient care is to actually do a blood-based test. Now, we're offering one way of getting hold of the cancer cells, which just seems to be very effective. And that can change the whole paradigm. And if I just give you a financial thing, you have new immunotherapy drugs coming through which target improving the immune system's response to cancer. Those drugs cost £150,000 a year, and they only work for 1 in 3 patients. So with 3 blood tests, we can save £300,000 by telling you who's the one who's going to benefit.
So what's holding them back from signing up?
What's holding us back? High-quality evidence, data. So we need clinical trials done by these leading cancer centres, and we also need regulatory approval, which we have in Europe with the CE mark, and we're working hard on to get FDA in the United States. You
also talked about that you're looking for customers. I know you've got some conversations that are happening behind the scenes, but where are those customers? Where are your ideal customers coming from?
We're limiting our customer base at the moment to major countries in Europe and the United States. And we've got several customers who are evaluating the product at the moment, and hopefully we'll be making some sales decisions fairly soon.
When you say fairly soon, are we talking...
Well, I promised that we'd do it by the end of this year, and I'm hopeful that we'll meet that promise.
OK, you've only got 33 days left.
Sorry?
I think 33 days left to the end of the year.
There's only a few days left.
Working days, not sure. Right, any questions from you? Yeah, second row, please, and we'll come to you afterwards. Yeah.
Thank you very much. Interesting to hear how your business is progressing. Is the sale of devices to the trials market going to be sufficient to generate income that will turn you into a cash positive business, or do we have to wait till the main use of the device for monitoring it becomes the market?
Yes, so we did a fundraise in February this year, which raised money for 2 years. So we got 2 years' money, and that was very specific for the ovarian cancer clinical application and for the research use sales. And yes, we have enough money to do that. In terms of whether the research use market will generate enough money for all of the development work we want to do in the future, it depends on how much development we choose to do. So if we expand into other clinical applications, then more than likely that wouldn't be enough. If we don't, if we keep with the ovarian cancer and the research use, then yes, it should be sufficient. Thank you.
There's a question towards the back.
Who are your main competitors?
There's several categories of competitors. There's one major category which is not circulating tumour cells, which I referred to, which was cell-free DNA, or otherwise known as ctDNA. And what these companies are doing is they're looking for DNA fragments in the blood, not intact cells, but when a cancer cell dies, you get a fragment of DNA in the blood. I pointed out that Cancer Research UK had made our system completely compatible with that alternative approach. You can use the same blood sample both for cell-free DNA and for circulating tumour cell analysis. And that's important. But the other thing is circulating tumour cells, because they're live cells, you get a lot more information, not just DNA, you can get RNA and you can get gene expression. So that's where I think the biggest issue is, and I want to position this so that anybody who wants to go down that route would automatically say, well, if I'm doing the work, might as well do Parsortix and get the circulating tumour cells as well. So that's that plan. There are other companies developing circulating tumour cell systems, but none of them, obviously, because we own the patent, they haven't got the step-based approach. The main leading alternative is in antibody-based capture, and that has some certain problems and doesn't capture all the types of cells. And that, again, is why I'm pleased that Cancer Research UK Manchester had done this huge study, and they compared us against the gold standard and various other things as well. Thank you.
2 questions behind you.
Andrew, at the start of your presentation, you mentioned the types of cancer that have a specific signature, you know, the HER2 and the whatever the other one's called. It seems that most cancers, the molecular signature is a great deal more complicated, and there are all sorts of different mutations and so on. Is that going to make it really almost impossible to find targeted cancer drugs? And, you know, I know that's really not really your problem, but I mean, do you see it as a problem for, you know, the treatment of cancer in the future?
You're right that it's outside of my sort of technical competence to talk about the development of new drugs. However, it is definitely the fact that these big companies are developing hundreds of potential candidates for that. Cancer Research UK Manchester have got a plan to have 50 new compounds going through their own research institute in clinical trials every single year. Now, obviously, early stage trials, a lot of them fail, but many won't fail. And they want to use our system to actually work out which patients should go in which trial. So I guess the more complicated it is, the bigger the challenge for a new drug, but you need a system like ours to get hold of the cells to do the analysis in the first place.
I mean, sorry, I was interested that in your one of your projects, you're able to pick out 30 RNA markers, so potentially you could pick out hundreds of 30.
You could pick out hundreds, and if you had a multiplex system, you know, we might choose to have 30 in our final product. Probably we'll reduce it down to a smaller number, and that's what we're working on at the moment in terms of a training study. A smaller number which is accurate would be good, but if you have to do a larger number, you can do.
Okay, we've got time for 2 more questions. One there, and then we'll come to you after.
Hi, Andrew. Just in terms of an FDA update, I think previously you've given an update saying that you're about 2/3 through the list of questions that were posed by the FDA. If you could perhaps just give us an update on that piece, please. And just secondly, about the ovarian trial, I think you mentioned that the US leg was you're hoping to complete that 6 months later. Does that imply implicitly that you will have FDA sort of 6 months, so the first half of next year? Is that what you're aiming towards, please?
Just to put the context to the question, there's no company that has FDA authorization for cancer cell harvesting and analysis. There's one company that has FDA for cancer cell capture and counting, but they can't get the cells out for analysis. So what we're trying to do is something that's never been done before, which is get the FDA to approve a simple system for taking cancer cells out of the bloodstream. We're trying to keep it as a wide application. We call it a platform application because we don't want to be specific about what the end user does with the cells, because the wider use of those cells, the better from our standpoint. We have, over fairly recent times, we've actually had additional meetings and other interactions with the FDA. We've been through in very great detail the information we've provided them, how we think it meets their questions that they raised, what we consider to be their remaining questions, and how, in great detail, how we'll do studies to meet those requirements. We continue to get a favourable response from the FDA. But they do ask for some very, very challenging things to be proven. So, for example, we need to demonstrate that our system works well, even dependent on what the medication that the patient is having. So we have to take those medications and we're going to have to actually put them in the blood to make sure that we can still get the system to work. We need to be able to prove to them how long we can hold the blood for before we separate it. We need to prove that 3 different cancer centres in different cities can get the same result with the same patient's blood, and. So there's lots of different things that they want us to prove about the analytical performance of our system and the clinical performance. Those are things that we believe we can do, and the things that we haven't, we've done a lot of them. The things that we haven't already done, we've got a very clear plan, and they'll never actually endorse or approve it, but they haven't said they've got a problem with it. So we feel that we're moving in the right direction with FDA, but I can't give any timescale on when that will be completed. The second part of your question was about the ovarian cancer application. And so understanding, of course, that the FDA may take longer in some circumstances than we'd like, we've made sure that the ovarian cancer clinical study is happening in Europe as a standalone entity. We estimate that's going to be, there's 50 patients in the training study, then another 250 patients of women who are about to have surgery for an abnormal pelvic mass. So these are, and then benign versus malignant. And then we expect to have another 150 patients in the United States, which that one won't be standalone. That one will build on top of the evidence from Europe. And we're building into the study plan interaction with the FDA throughout the process so that they see the study plans before we actually start them in the States. So hopefully that will mean that our estimate is a further 6 months before we can complete in the US.
And there was that last question of the back row.
Thank you. Four very easy questions. First of all, are you on the AIM market?
Yes.
Secondly, what price are the shares?
They're about 76.5p the last time I looked.
Oh, not bad.
And your question?
Yes, there was... I'll see you outside. I've forgotten it for the moment.
Well, you'll be around afterwards to catch
Yeah,
up.
I'm around.
But,
I'm
but
happy
what I did want to say is, we started off the day talking about how biotech is off those great, the highs that it was maybe 3-4 months ago.
Yeah.
Has the bubble burst?
Has the bubble burst? Well, we hear stories about how the US market's coming back a bit from biotech side with new pharmaceutical drugs, and Hillary Clinton saying she's going to squash down the prices, etc. I actually think that Angle's and our Parsortix system is part of the solution, because, as I said, there's so many expensive drugs being prescribed to patients who don't even benefit from them. So the best way to save money is to discriminate which drug will help the patient and which one is irrelevant for them. So that would be my focus on that question.
Well, Andrew Newland, thanks ever so much. It's been a real pleasure hearing from you, as it has hearing all of the other people here in the audience. I mean, precision medicine, that's what we've been talking about.
Precision medicine.
We're talking about revolutionising cancer care, stroke care, diabetes care. We've had some really interesting, insightful speeches this morning, haven't we? Have some of the drugs we've heard about, have they got blockbuster potential? Well, that's probably for you to analyse and work out, going through these presentations with a fine-tooth comb. But also, you've seen the brains behind these businesses. You've heard from them. You've seen the twinkle in their eye. So you can come up with those decisions yourself. You never know. We might have a blockbuster drug here. So I'd like to thank you. Thanks ever so much. Andrew, thank you. Michael from Uneuron, Cameron from Volition RX, Ewan from Deltex, Tim from Sarium, and Craig from C4X Discoveries. A big hand for everybody. And of course, don't forget, lunch will be served, a chance to network. So do hang around for that as well. And our next event is going to be Mining Capital Conference. That's going to be here. Same place, same time on the 7th of December. Thanks very much.